In this video, we will discuss about the mass spectrometry analysis of tumor-derived lymph node from different chemogenetic model. So basically, we are continuing our discussions where we have stopped in our previous video. So in this video, they have analyzed, or the results that are shown in this video reflects the mass spectrometry analysis of this tumor-derived lymph node. So they have compared the tumor-derived lymph node with one model that caused tumor suppression and the other model that caused tumor progression. So in the tumor suppression model, they have checked or presented the results for the different immune cells. So in terms of the different immune cells, the immune cells that are important for our analysis is the CD8 plus T cells. So it shows that in the presence of their HM4 DGI model, there is an increase in the CD8 plus T cells, and also there is an increase in this gamma delta T cells. And at the same time, these conventional dendritic T cells are also increased. And this is for the model where HM4 DGI is used, and this model causes tumor suppression. And in the other model that promotes the tumor progression or helps in the tumor evasions, which is related to the optogenetic models, optogenetic model which promote this calcium within the system. And this model shows it is completely reverse results. That is, it shows a decrease in the CD8 plus T cells, decrease in this gamma delta T cells, and also decrease in this conventional dendritic C cells. And this is expected as the immune evasions environment usually have the lower amount of or have the less amount of activated T cells. And thereafter they checked this different variants of different immune cells in the mice that are injected with different tumors, that is SCC7 and B16F10. And the results for in the panel H shows for the model where they use HM4 DGI or the models that cause tumor suppressions. So in this case, they are able to see increase in the CD8 plus T cells for both the system, gamma delta T cells, and also activated conventional dendritic C cells. So this shows that the results that they are seeing is not just specific to a particular kind of tumor cells, but it can be expanded to the other tumor cells. And when they checked the combinatory effect of this two model, that is when they combine the tumor progression model, that is optogenetic based model, and the tumor suppression model, that is chemogenetic based model, they want to see what is the overall results that they get. And in that case, they are able to see that when both the models are combined, they are able to see enhanced CD8 plus T cells, gamma delta T cells, and also activated dendritic conventional dendritic T cells. This indicates that the tumor or chemogenetic inhibition ability overcomes the tumor suppression ability of this optogenetic based model that cause this calcium increase within the cells. Thereafter, they wanted to confirm whether this, in the case of chemogenetic inhibitions, the tumor cell killings are done by the dendritic cells or there is some other factors. So they focused on first checking whether the dendritic cells plays a role towards this tumor cell killing. So for that, they use ZsGreen labeled tumor cells and checked this conventional dendritic cells. They are expected that if the dendritic cells goes under phagocytosis, then it will kill the tumor cells or it will engulf the tumor cells. So they will also have the ZsGreen fluorescence. So when compared to the control, they are able to compared to the control where they don't have ZsGreen tumors, they are able to see enhanced signal for the ZsGreen for enhanced signals for the ZsGreen for both the tumor cells infected mice, indicating that the conventional dendritic cells are the one that takes place. Conventional dendritic cells are the plays an important role, or plays a role towards killing of the tumors in the chemogenetic inhibition method. After that, they checked it with other parameters also, where they checked the release of the granzyme, which is the lytic granules, and the growth factors, or the growth factors such as IFN-gamma and TNF-alpha, which also cause cell death, which also helps in the, which are also released and help in the tumor cell killing. And so this experiment is done with this CD8 plus T cells that are derived from tumor derived lymph node, and co-culture them with those tumor cells in the presence of this HM4 DGI, or this tumor cells has this HM4 DGI. So they are able to see that in the presence of chemogenetic inhibition method, there is a increase in the granzyme and IFN-gamma and TNF-alpha compared to the control, indicating that this tumor-derived dendritic cells, or these CD8 plus T cells that are present in the tumors, plays a role towards this tumor cell killing. So overall, it's the tumor infiltrated lymphocytes that enhanced, or that has a role towards this tumor cell killing in the presence of chemogenetic inhibition method that they have tested and developed and tested. So that's all for this video. We will discuss other results in our subsequent videos. So thank you for your time. And if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description.